Hello guys, this big topic is physiology of bacteria. It is the microbiology topic and this is most important question in previous year question paper. First, we will see about physiology of bacteria. The learning objectives are bacterial growth and nutrition, factors affecting growth of bacteria and bacterial metabolism. These are the learning objectives in the physiology of bacteria. First, we'll see bacterial growth and nutrition. In even if it is the learning objective of the physiology of bacteria, it contains some other subtopics that are all bacterial growth requirement, bacterial vitamin, bacterial cell division, rate of multiplication in bacteria, bacterial count, bacterial growth curve. That's it. First, we will see bacterial growth requirement. Bacteria contain 80% of water, so it requires just minimum amount of nutrition. That the nutrition is one required for growth and multiplication. The nutrition consists of carbon, hydrogen, nitrogen and oxygen like that us. And even it wants some other special nutrition like sodium, potassium, sulfur, magnesium, manganese, iron. And coming to bacterial vitamin is that means it wants special nutrition to grow in culture media. That means some other bacteria don't like to grow in normal culture media, so it wants special some certain organic compounds in culture media. So that's called bacterial vitamins or growth factors. Even it is common with mammalian vitamins. For example, if it is some bacteria grow in vitamin B12 and pyridoxine, nicotinic acid and riboflavin. Example, riboflavin is bacterial vitamin for Bacillus anthracis and pyridoxine is bacterial vitamin for Lactobacillus species. And coming to bacterial cell division. Even bacterial cell division is not a multi complex form. It's just a simple form. That is binary fission. It means a single mother bacteria can divide or can give two daughter cell bacteria. That's it. So in this method, it have nuclear division and cytoplasmic division. In nuclear division means bacteria contain two strands of bacterial DNA and it is separated and replicate and it form new complementary strand. These two identical molecules of DNAs, double stranded DNAs again formed. And cytoplasmic division, the cytoplasmic division occurs in like transverse septum. It grows across the cell from cell membrane following the cell wall material or deposited and two newly formed cells are separated. That's it. And coming to rate of multiplication in bacteria. In this rate of multiplication in bacteria is generation time. Generation time means time required for bacterium to give rise to two daughter cells. It's different for different bacteria. Example, uh, if E. coli wants to give two daughter cells, it will take 20 minutes. Mycobacterium tuberculosis take 10 to 15 hours to give two daughter cells. But in here see geometric progression. A single bacteria can theoretically give 10 power 21 daughter cells. But functionally that's not happen. because lack of nutrition and toxic accumulation leads to bacterial death, or it can't produce further daughter cells. And coming to bacterial count, this is very easy. That the bacterial count is, we can further divide it into total count and viable count. In total count means living cells and died cells. In counting chamber we can count by microscope. Okay, total count means living cells and die cells. But in coming to viable count, living cells only can counted by four plate method. And coming to bacterial growth curve. Bacterial growth curve is important for microbiology. We should know because uh, if we go in lab for how the culture media works and what are all requirements for culture media to grow, we should know the bacterial growth at time. First, in bacterial growth curve, when bacteria inoculated in suitable liquid culture medium and incubated, its growth follows some courses. First, lag phase, log phase, stationary phase, and decline phase. See here, this is lag phase. In this lag phase, the bacteria immediately start growing or divide. In the lag phase, the enzymes are produced and synthesized, and in the log phase only it starts dividing immediately and utilize the enzymes and metabolites to divide the cells. And in the stationary phase, the number of progeny cells are only divided, and in the stationary phase the enzymes and metabolites are uh, deficiency. And in the phase of decline means uh, it reduce the cell division, and even it is completely stop in the cell division, bacterial cell division in the decline phase. First, one by one, see. In the lag phase, the period between the inoculation and the beginning of multiplication of bacteria. After inoculating into a culture medium, bacteria don't start immediately multiplying. It takes time for build up enzymes and metabolites. So the bacteria increase in size due to accumulation of these enzymes and metabolites, and bacteria reach maximum size at the end of the lag phase. And coming to log phase, in the log phase only the bacteria can start divide, so smaller in size the bacteria. And in this lag phase only biochemically active, because the best stage to perform biochemical reactions, and uniformly we can stand in this log phase. And coming to third phase, the stationary phase. After the log phase, the bacterial growth ceases due to exhaustion of nutrition and accumulation of the toxins. The number of progeny cells are formed. It's enough to replace the number of cells that died. The number of viable cells remains stationary, has almost a balance between dying and newly formed cells. The total count keeps rising in this stationary phase. Back in this phase, bacterium become gram variable. More storage granules are formed and sporulation occurs. Bacterium produce exotoxin, antibiotics and bacteriocins. In the fourth decline phase, Gradually, the bacteria stop dividing and completely. Cell death continues due to exhaustion of nutrition and accumulation of the toxins. We can here see the various phases of bacterial growth. In bacterial divide in the log phase, stationary phase, decline phase, then there is no bacterial division, and the lag phase also there is no division. In the bacterial death, only in the decline phase and stationary phase. In the total count, in the lag phase is very flat, in the log phase rise, stationary phase also rise. In the decline phase, the total count is flat. In viable count, in the lag phase it's flat, and log phase it's rise, and the stationary phase flat, and the decline phase it's falls. The special features between the four phases. First, in the log phase, accumulation of enzymes and metabolites and attain maximum size. In the log phase, the uniformly stained, metabolically active and smaller in size. In the stationary phase, the gram variable. It produces granules, spores, exotoxin, antibiotics, and bacteroidein. In the decline phase, it produces involution forms. Coming to factors affecting growth of bacteria, that's oxygen, carbon dioxide, temperature, pH, light, osmotic effect, mechanism, and sonic stress. These are all factors affecting growth of bacteria. And also, first, oxygen. Some bacteria grow only in the presence of oxygen, that is called obligate aerobes. For example, Pseudomonas, Mycobacterium tuberculosis, and Bacillus. These are all obligate aerobes. And obligate anaerobes, they grow absence of oxygen. So obligate anaerobes. Example, Clostridium tetani. And third, facultative aerobes. They are anaerobes, but they can also grow aerobically. Example, Lactobacillus. And fourth, facultative anaerobes. They are aerobes, but also grow in anaerobically. Example, E. coli. And fifth, microaerophilic bacteria grow in presence of low oxygen tension. For example, Helicobacter, Campylobacter. And coming to 6, aerotolerant anaerobes. They can tolerate oxygen for some time, but it doesn't use it for it. For example, Clostridium histolytica. And carbon dioxide, next factor, higher amount of carbon dioxide for growth it use, that's called capnophilic bacteria. For example, Brucella abortus, Streptococcus pneumoniae. And coming to temperature, most of the bacteria grow optimally at 30 degrees Celsius, but it differs for bacterial species. Psychrophiles, it's below 20 degrees Celsius it only can grow. Example, Pseudomonas. And second, Mesophilus. The range between the temperature is 25 degrees Celsius to 45 degrees Celsius. Example, most of the pathogenic bacteria can grow in this Mesophilus. And coming to Thermophilus, high temperature, 55 to 80 degrees Celsius. For example, Geobacillus, Styrothermophilus. And pH. Most of the bacteria grow between 7.2 to 7.6 pH. That means it's basic. At acidic pH below 4, some bacteria grow. Example, lactobacillus. At alkali pH above 8, some bacteria can grow. That's it. Example, Vibrio cholerae. The next factor is light. Bacteria grow well in darkness only. It's sensitive to ultraviolet rays and radiation in light. But photochromogenic mycobacterium produce pigments when exposed in lights. And coming to osmotic effect. If bacteria sudden exposed to hypertonic saline solution, the cell shrink. That's plasmolysis. At the same time, when the bacteria is exposed to hypotonic solution or water solution, that is the bacteria swell and rupture. That is called plasmoptosis. And the next factor is mechanical and sonic stress. Bacteria have a tough cell wall, may rupture and disintegrated by vigorous shaking with the glass beads and exposed to ultrasonic vibration. The next factor is moisture and dedication. 80% of bacteria cell consist of water. It's minimum requirement for growth. We already see some organisms like Trypanosoma pallidum and Necessary Conariae die after drying. At the same time, some bacterium like Mycobacterium tuberculosis may survive drying for several weeks. Drying in cold and wet come. This is used for preservation of bacteria. The final is bacterial metabolism. Bacterial metabolism is the process by which a microbe obtains energy, nutrition for survival, and reproduction. It's based on three principles: how the bacteria obtain carbon, how the bacteria obtain electrons, and finally, how the bacterium get energy. First, how the bacterium obtain carbon for synthesizing cell mass. We will see autotrophs and heterotrophs. In autotrophs, bacteria can synthesize all organic compounds by using atmospheric carbon dioxide as a source of carbon. Heterotrophs. They used reduced and performed organic molecule as a carbon source. And coming to how the bacteria obtain reducing equivalents, that means electrons, used either in the energy conservation or biosynthetic reaction, that is lithotrophs and organotrophs. Lithotrophs obtain reducing electrons or equivalents from inorganic compounds. In organotrophs, it's obtained reducing equivalents or electrons from organic compounds, that is called organotrophs, and lithotrophs from inorganic compounds. And coming to third, how the bacteria obtain energy for living and growing, that is hemotrophs and phototrophs. In hemotrophs, obtain energy from external chemical compounds. In phototrophs, obtain energy from light. Bacteria usually possess these properties. Most of the bacteria contains hemo, organo, heterotrophs. For example, E. coli. That's the best example for hemo organo heterotrophs. That's it. Thank you.